Assalamu alaikum student. Today the lecture is about the factor affecting rate of enzyme action. You know that thousands of amino acids coiled upon themselves to form a globular structure of an enzymes. Different amino acids are present in enzyme to form a globular structure of enzymes. They coiled upon themselves like that and form a globular structure of enzymes. The structure or interactions of enzymes is very important for its proper functioning. In living cell, many factors can influence the catalysis of enzymes. First one is temperature and the second one is pH, third one is substrate concentration and fourth one is enzymes concentration. First one is temperature. Let's study one by one. Temperature, the enzymes work to its maximum rate at a specific temperature which is called optimum temperature. Each enzyme is most active at a particular temperature. Starting with 0, the kinetic energy of molecule increases with the rise in temperature. More molecules collide and undergoes a reaction. For example, the optimum temperature present in human body is 37 degrees Celsius, and increase in temperature also speed up the rate of chemical reactions or enzymes catalyzed reaction, but it happens only to points. As our temperature is increased to a certain limit, heat energy contribute to the activation energy, also provide kinetic energy to the reaction, and the reaction is speed up. However, heat energy increase the vibrations of enzyme atom when the temperature is elevated above the optimum temperature, and that result in the loss of globular structure of enzyme. So this process is called enzyme denaturation. Denaturation of enzyme reduces the conversions of substrate into product and affect the enzyme catalysis of reaction. After crossing the optimum temperature, enzymes activity decline, and since the bond in the protein molecule are disrupted. At time, high temperature result in denaturation of enzymes molecule. More for most enzymes, optimum temperature is lies between 35 to 40 degrees Celsius. At low temperature, enzyme reactions require more time. Next one is the pH. Effect of pH. The second factor that affects the rate of enzyme reactions is pH. All enzymes work to their maximum rate at a small range of pH, which is called optimum pH. A minor change in this pH induces denaturation or complete blocking of enzymes catalysis. Each enzyme has its own optimum pH, which is unique to it. For example, pepsin, which work in the stomach, is active as acidic pH from 2.3 to 5.2 to 3.5, while trypsin enzyme, which present in the small intestine, is an acidic work as alkaline pH, which range from 6 to 8. Now the amino acids which are present at the active sites are affected by change in pH, and that is why the enzyme lost its ability to catalyze substrate. Most enzymes have characteristics pH at which their activity is maximum. Above and below the pH, the enzyme activity decreases. If a curve is drawn between enzyme activity and pH on a given substrate concentration, maximum activity is revealed as a defined pH known as optimum pH. The third one is substrate effect of substrate concentration. The initial velocity of an enzyme catalyzed reaction is proportional to the substrate concentration. कि एंजाइम्स कैटालाइज का जो रिएक्शंस होता है वो प्रोपोर्शनल होता है सबस्ट्रेट कंसंट्रेशंस के विद द राइज ऑफ सबस्ट्रेट कंसंट्रेशन. जैसे ही हम सबस्ट्रेट कंसंट्रेशंस को इंक्रीज करेंगे तो एंजाइम्स की एक्टिविटी भी क्या होगी, सर्टेन पॉइंट से इंक्रीज हो जाएगी. आफ्टर दिस पॉइंट a further increase in substrate concentration has no effect since the enzymes will saturate. Substrate concentrations and increase of substrate concentration increase the rate of enzyme catalysis reactions. If the concentration of enzymes kept constant, अगर constant रहेगी enzymes की concentration, then substrate activity increase a point or reached where the rate of reactions is no longer increased by any substrate increase in the substance. The reason behind is that all the enzymes they are occupied in catalyzing the substrate and there is no free active sites present which can accommodate the incoming substrate. This state is called saturation of active sites and the rate of reaction remains stable and it does not increase. The fourth and last one is enzyme concentration. The concentration of enzyme is a specific time in the presence of unlimited substrate is very important for the rate of reaction and increase the amount of enzymes by twofold can increase or double the rate of enzyme catalyzed reactions. An increase in the enzyme concentrations mean an increase in the number of active sites. So more active sites means more conversions of substrate into the product. So this is all about the factor affecting on enzyme concentration. Allah Hafiz.